Hello, this is Dr. Morris. I'm continuing with the lecture series for Anatomy and Physiology 1. This will be an introduction into chemical and cellular level organization. The previous lectures have detailed the chemical makeup of the human body, starting at an anatomical and then a molecular level. We'll now go into some of the chemical and cellular level organization aspects of the human body. At a cellular level, the human body is made up of several trillion units. These units are called cells, vary widely in size and appearance. They perform many functions in the body. Some of them are specialized functions and vary significantly depending on the locations of these individual cells. The smallest entity that can perform all the basic life functions are the cells. Cells in the human body depend on one another to carry out these specific functions, and the actions of one cell can affect all the other cells. Now, we've already discussed the aspect of homeostasis in the initial lecture of the semester, but let's review. Homeostasis: cells work together in relative harmony, coordinating their activity. Cell has a particular role to play, and cells help maintain the body's homeostasis, otherwise referred to as the steady state or the balance inside the human system. Historically, cells were studied or have been studied microscopically. A typical cell measures approximately 0.1 millimeter in diameter. Microscopes were invented back in the 17th century. Dr. Robert Hooke in 1665 inspected thin slices of cork as if you'd fish with, and found that several of them consisted of millions of small irregular units. Concepts of the cell theory were formed based upon these observations. The two most common methods used in studying cells in tissue is referred to as the light microscopy or electron microscope. The cell theory, which I alluded to earlier, basically cells are the building blocks of all plants and animals. All cells come from the division of pre-existing cells. Cells are the smallest unit that can perform all vital physiologic functions. Each cell maintains homeostasis at a cellular level, and then homeostasis at the level of the tissue organ or organ system, and then the organism reflects a combined and coordinated action of many cells. A major difference between the two techniques is the level of resolution. Light microscopy permits us to see individual cells and the largest of the internal structures. Electron microscopy, however, allows us to discern the smallest internal structures within the cells. Your textbook gives some illustrations of light microscopy and electron microscopy, and as we go further into this lecture, I'll have some illustrations and pictures for you as well. Some areas of cell anatomy that are important to at least recognize the definitions for. Cytology basically is the study of cellular structures and functions. Cell biology incorporates aspects of cytology, biology, chemistry, and physics. There are two classes of cells in the body: sex cells, also known as germ cells or reproductive cells. Sex cells are either sperm for the male or oocytes for the female. Fusion of sperm and oocytes at fertilization is the first step in the creation of a new individual. Somatic cells, soma, referring to the body of the cell, include all other cells in the human body. This is an illustration of a typical cell. It has been modified a bit to demonstrate some of the surface appendages that you can see existing on cells. For example, cilia are structures that we will talk about, finger-like projections, and even smaller finger-like projections referred to as microvilli, which are typically used to increase the surface area. But again, we'll go into more detail as we get into the specifics of these structures and organelles. And then also, if you look at the internal environment and the organelles or little structures that are located inside the cell, you'll see there are many names, many structures, each of which you'll have to become familiar with to understand not only the structure but also the functioning of each of these individual components. The extracellular or interstitial fluid compartments, cells are surrounded by a watery medium. This medium is known as extracellular fluid. Extracellular fluid in most tissues is called interstitial fluid. The cell membrane or plasma membrane or plasmalemma, this is the outer boundary of the cell that separates the cell content, the cytoplasm, from the extracellular fluid. The cytoplasm can itself be subdivided into cytosol, which is a liquid, or organelles, which is the intracellular structure. The cell membrane provides specific functions. One is physical isolation, separating what's on the inside of the cell from the outside environment, sensitivity, and then structural support. And we'll go into some more details about each of those characteristics. Physical isolation. The cell membrane is a physical barrier that separates the inside of the cell from the surrounding extracellular fluid. Conditions inside the cell and outside the cell are very different. And those differences must be maintained to preserve, again, that key word, homeostasis. Examples: the cell membrane keeps enzymes and structural proteins inside the cell. The cell membrane controls the entry of ions and nutrients such as glucose, elimination of waste products, and the release of secretions. As for sensitivity, the cell membrane is the first part of the cell affected by changes in the composition, concentration, pH, and extracellular fluid. It also contains a variety of receptors that allow the cell to recognize and respond to specific molecules in its environment. For instance, cell membrane may receive chemical signals from other cells. The binding of just one molecule may trigger the activation or deactivation of enzymes that affect many cellular activities. Structural support, specialized connection between cell membranes and other membranes and extracellular material give tissue a stable structure. An example: cells at the surface of the skin are bound together while those in the deepest layers are attached to extracellular protein fibers in underlying tissues. Structural support, specialized connections between cell membranes or between membranes and extracellular material give tissue a stable structure. An example of that: cells at the surface of the skin are bound together while those in the deepest layers are attached to extracellular protein fibers and underlying tissues. Composition of cell membranes. A cell membrane is extremely thin, delicate, ranging from 6 to 10 nanometers in thickness. This membrane contains lipids, proteins, and carbohydrates, structures of which we have discussed in previous lectures. The phospholipid bilayer. Lipids form most of the surface area of the membrane. Cell membrane is called the phospholipid bilayer because the phospholipid molecules in it form two layers. A hydrophilic end, a definition you should be familiar with at this point from previous lectures, contains the phosphate portion, and then the hydrophobic end, or the lipid portion, the phospholipids lie with their hydrophilic heads at the membrane surface and their hydrophobic tails on the inside. This is an illustration taken from your book demonstrating the phospholipid bilayer. You should become very familiar with this picture because the concepts that you can take from this picture will help you to understand phospholipid bilayer, the phosphorus end, the hydrophilic end, how the environment outside the cell is different from the environment inside the cell. Being able to label each of the components that we see in this cell membrane will also help you to appreciate the functions of carbohydrates, proteins, and lipids. Just a quick view on the picture demonstrates to you first two environments. The environment outside the cell, referred to as extracellular fluid, outside the cell, and then the environment inside the cell, referred to as cytoplasm or cytosol. Again, these two environments are very different. This is the cell membrane. and the cell membrane is composed of a phospholipid bilayer. The phosphorus ends have contact with an aqueous medium. The extracellular fluid contains a significant amount of water, so phosphorus can interact with water, thus the hydrophilic portion of the cell membrane. Also, facing the internal environment, phosphorus there as well. Why? Because this is also an aqueous environment. Though the aqueous environment here is grossly different from the aqueous environment on the outside, because it's an aqueous environment, it has to be able to communicate with the cell membrane. Thus, the phosphorus, or the hydrophilic ends, have to be able to communicate with the internal environment. Then facing the inside of the cell membrane are the lipid tails. This gives thus the name phospholipid bilayer. There are two layers of lipids here, the tails from the phosphorus structure and the lipid on the inside, and then again phosphorus end here, lipid end here facing the inside. The lipid bilayer prevents water and other aqueous solutions from freely passing through the cell membrane. If the environment from the outside invaded the inside, the cell would die. If substances inside the cell would freely float to the outside, the cell would die. So the lipid bilayer prevents that free movement of the environments and thus separates the two environments. You can also see the cholesterol that's stacked inside. Remember, cholesterol derived from lipids. Also, you see protein structures. We'll talk more in detail about proteins and their function in the cell membrane. But for now, look at the proteins simply as channels, ways for substances outside the cell to be able to get in. Remember, the cell requires nutrients, so substances, including water, has to be able to get into the cell for it to be able to survive. But it's regulated by these protein channels. We also see carbohydrates in the form of the glycocalyx or glycolipids that exist on the surface of the cell. We'll go into a lot more detail about each one of these structural components, but that's just to give you a brief overview of the phospholipid bilayer, again, the cell membrane in the human cell. The phospholipid bilayer, again, has a hydrophilic head of two layers that are in contact with aqueous environments on either side of the membrane. The interstitial fluid on the outside, interstitial fluid also, as I've mentioned to or referred to on the picture as the extracellular fluid on the outside, and then there is the cytosol or the cytoplasm that's on the inside of the cell, and the hydrophobic tail forms the interior or the center of this phospholipid bilayer. Ions and water-soluble compounds cannot freely enter into the interior of the cell. Thus, water is blocked. Because lipid tails and the phospholipid molecules are hydrophobic, will not associate with water molecules. Water and solutes cannot cross the lipid portion of the cell membrane. Again, as the illustration shows, this is the lipid membrane. This is the lipid portion of the membrane. Again, water cannot freely communicate here. Water from this side can't freely communicate outside. The only way water and other substances can pass through here are basically through these protein channels. Now, I said only. There are some exceptions to that, and we'll talk about those more and more as we get into the lecture series. But for right now, I just want you to appreciate if substances are going to get into the cell, typically they have to come through these channels. The term hydrophobic compounds in the center of the membrane isolate the cytoplasm from the surrounding fluid environment. Isolation of the cytosol. Such isolation is important because the composition of the cytoplasm is very different from that of the extracellular fluid, and the cell cannot survive if the differences are eliminated. Proteins. Proteins are denser than lipids. They account for roughly 55% of the weight of the cell membrane. There are two general classes of membrane proteins. There are integral proteins, and then there are peripheral proteins. Integral proteins are part of the membrane structure. They cannot be removed without damaging or destroying the membrane. They span the width of the membrane one or more times, and then because of that, because they span the width of the membrane, they are referred to as transmembrane proteins. They're bound to the inner and the outer surface of the cell membrane and are easily separated from it. They greatly outnumber peripheral proteins. Anchoring proteins basically are membrane proteins. They attach the cell membrane to other structures and stabilize its position. They're bound to the cytoskeleton, a network of supporting filaments in the cytoplasm. Outside the cell, they may attach the cell to extracellular protein fibers and to one another. There are also recognition proteins or identifiers. Cells of our immune system recognize other cells as normal and abnormal due to the presence or the absence of characteristic recognition proteins. In other words, for our immune system to be able to recognize our own cells as a part of our body and not an invading cell from, say, a microbe or a virus, the only way that our immune system can recognize our own cells is because of specific recognition proteins or glycoproteins, which is a combination of carbohydrates and protein. These are recognition proteins or identifiers. Without those identifiers, our immune system will attack our own cells and destroy our own cells. So it's a very important characteristic. Now, enzymes. Integral or peripheral proteins. Enzymes catalyze reactions in extracellular fluid or in the cytosol depending on the location of the protein and its active site. Now, we've already discussed proteins, we've already discussed enzymes, we've already actually discussed active sites as well. But it's important to understand the concepts of these and now their roles inside the developing cells. Dipeptides are broken down to amino acids by enzymes, which is a great example to use. Exposed to membranes of cells that line the intestinal tract. There are also receptor proteins, or ligands. The cell membrane are sensitive to the presence of specific extracellular molecules. These specific extracellular molecules are referred to as ligands. They can be anything from small ions like calcium or relatively large complex hormones. But again, ligands can be anything from small ions to relatively large complexes. And these are molecules that are specific extracellular molecules. Receptor proteins are exposed to the appropriate ligand, and they'll bind it. And by binding it, it will trigger the changes in cellular activity. The binding, an example would be the binding of the hormone insulin to a specific membrane receptor. Protein is the key, which will lead to an increase in the rate of glucose absorption by the cell. So this is just one example of the ligand and receptor relationship. Cell membranes differ in the type and the number of receptor proteins that it contains. Differences account for different sensitivities to hormones and other potential ligands. Carrier proteins bind solute and transports them across cell membranes. The transport process involves a change in the shape of carrier proteins. The shape changes when solute binding occurs. The protein returns to its original shape and the solute is released. Carrier proteins require energy. Carrier proteins may require ATP as an energy source. All cells have carrier proteins that can bring glucose into the cytoplasm without expending ATP. Cells must expend ATP to transport ions such as sodium and calcium across the cell membrane and out the cytoplasm. Channels. A central pore or a channel forms as a passageway completely across the cell membrane, permits the movement of water and small ions, solutes across the cell membrane. Ions do not dissolve in lipids, so they cannot cross the phospholipid bilayer. Ions and other small water-soluble material can cross the membrane only by passing through the channels. Now the channels, as I alluded to earlier, were protein structures, integral protein structures that were in the illustration. They lie and they pass through transmembrane, pass through the cell membrane. Many channels are highly specific, that is, they permit the passage of only particular ions. They also are extremely important in the physiological processes like nerve impulses, transmission, muscle contractions. Carbohydrates, another component of the cell membrane, 3% of the weight of the cell's membrane. Carbohydrates in the cell membrane are components of a complex molecule such as proteoglycans, combination of protein and carbohydrates, glycoproteins, combination of carbohydrates and proteins, and glycolipids, carbohydrates and fats. Carbohydrate portion of large molecules extend beyond the outer surface of the membrane, forming a layer known as the glycocalyx. If you go back to that illustration again, you'll see these carbohydrate structures that are projecting off the surface of the cell membrane consistent with the glycocalyx. What are the functions of the glycocalyx? And you should be familiar with these functions. 1, lubrication and protection, viscous layer that lubricates and protects the cell membrane, anchoring or locomotion, anchoring of the cell in place, also participates in the locomotion of specialized cells. The other things, specificity and binding. It functions as receptors, binding specific extracellular compounds such binding can alter the properties of the cell surface and indirectly affect the cell's behavior. Recognition, glycoproteins and glycolipids are recognized as normal and abnormal by cells that involve the immune response. And then again, I'll refer to the glycocalyx. As mentioned earlier, characteristics of the glycocalyx are genetically determined. Example is the presence or absence of membrane glycolipids on circulating red blood cells. For example, your blood type, type A, type B, type AB, type O. These are specific characteristics of the glycocalyx that helps your cells to be able to be recognized by our immune system. This is why it is so important that you are typed, your blood type, and you are cross-matched with individuals when you receive blood. If you receive blood from one of the groups that is not consistent with your own, your immune system will not recognize the glycocalyx on those cells, and thus your immune system will begin to attack the foreign cells. In attacking the foreign cells, the immune system will cause a severe inflammatory reaction. An anaphylactic reaction is the other name for it, causing the release of certain chemical substances in the body that can eventually lead to devastating health effects, if not even death. So it's important that the glycocalyx and the recognition factor for our individual cells is preserved. Glycocalyx and recognition, again, the body's immune system can recognize its own membrane and glycoproteins and glycolipids as itself rather than foreign. These recognition systems keeps our immune system from attacking our blood cells. They're still enabling it to recognize and destroy foreign blood cells. They should appear in the bloodstream. This ends this portion of the lecture. There will be a part 2 of the chemical and cellular structure lecture that will follow this one.